Hi guys, this is the last mini lecture on chapter 13. We've talked about how anatomy, fossils, and biogeography give us evidence for evolution. And this last section is on molecules, revealing relatedness. So it makes sense that if we could look at DNA of different organisms and see how similar or not they are in their genetics, the sequence of A and C and G and T, and if we could look at that and see how similar organisms are, it would give us one more clue. So I'm saying in this slide that it's highly unlikely that two unrelated species would evolve precisely the same DNA and protein sequences entirely by chance. They have to be related if they have such a similar sequence of DNA. That makes sense. This is an example from your text about looking at a particular protein. If we were to find a protein in lots of different animals and they all had that protein, and we could look for it in each one of them and see how similar those proteins were to each other, because those proteins are made from DNA, from a sequence of DNA, then it would give us a good idea of how similar or not these organisms are. So Cytochrome C is such a protein. Cytochrome C, all you need to know is that it's this protein from the mitochondria that is found in the chimp and the pigeon and the bullfrog and yeast and in all these organisms. So it's an easy lab test that we could do. We could collect that cytochrome C in all these organisms and see, is it really similar? Is the wheat similar to the human? Is the pigeon similar to the fruit fly? And we could compare it. So this chart shows the number of amino acid differences from humans. So if you got cytochrome C and looked at the DNA sequence of a human and then compared everything to that human, you would find that when comparing it to a chimp, the number of amino acid differences would be 0. They would be exactly the same. Now, if you just did a rhesus monkey, you would find one difference in that particular protein. You look at a rabbit, it starts to increase, 9 differences. A cow, 10, a pigeon, 12, and so on and so forth, till it gets to wheat. I mean, we still have that same protein, but it has considerable differences between what it looks like in a human and what it looks like in wheat. So think of it as one more tool. DNA is one more tool to look at different organisms and decide which are more related. Are we more related to a chimp or are we more related to a rhesus monkey? It's very useful for that. Look at this example. Here we have the common ancestor of a particular DNA sequence. Okay, any sequence that you pick of any protein, for example, like the cytochrome one we just picked. And you would have a sequence that went like this, for example, GACTTAGGGCT. That was 50 million years ago. And now, over time, there is a mutation. So 25 million years later, that's a long time later, this G here accidentally got a mutation and it turned into an A instead. So we switched something there and now it was an A. And then we let, another 25 million years go by and we still have that A switched, but we've acquired another mutation here, G. What used to be A is now a G. So we now have 2 mutations and it's giving rise to this modern species that now has this protein or this trait that has that particular sequence of DNA, and it lives happily ever after. The same thing could happen, which that A didn't change, but there was a mutation 25 million years later of this original common ancestor, but it was a different spot. It was this 4th here that used to be T, accidentally now is a C. And years go by, 25 million years go by, and that C still stays there in that organism, and yet we acquire one more mutation over time. Granted, it's taking a long time. So this organism today now has those 2 mutations and is also living happily ever after. Both of these, modern species 1 and modern species 2, had a common ancestor. It was this one. They are very similar in their sequence, but they do have some dissimilarities, right, that evolved over time. So that's how we think this DNA can help us by looking at these sequences and getting an idea of how similar or not these organisms are. In your book, you have a section called Investigating Life, and it talks about evolving backwards. We talked about the snake and the whale, and the cave salamander that's lost its eyesight because it's lived in caves for so long it doesn't need the eyesight. So you see sort of a leftover eye that's vestigial. So this is a nice breakdown of potentially the sequence of the whale going back to the water. So we believe that the common land ancestor, most common land ancestor to the whale, is the hippopotamus. And there were changes that went on in millions of years into an organism here called the Pakicetus. And this is a freshwater, semi-aquatic organism. So it goes in and out of the water, gets some change over many years, and ends up with another organism that's more in brackish water, which means it's semi-salty. And moving on to a salty habitat. Notice the streamlining now is happening. These quadrupeds, these legs now are disappearing. And we believe some sequence like this led us to then the modern day organisms that has both the baleen whales in it and the toothed whales, Odontocetes. So those in 2 different groups as modern organisms. So it's an organism, a really good example of having been terrestrial and having over time lost some of those characteristics that made you adapted on land and moving towards the water. End of chapter. Thank you.